robust hypotheses from ecology that can dictate whether or not there's limitations to dispersal to a site or overlap of niches competition that might regulate whether or not particular pairs of species can coexist in a particular site. But when we think about how often close relatives or congeners even can coexist, there's a little bit of additional information that's important here. There's also an addendum to these hypotheses called the limiting similarity hypothesis that suggests that because close relatives might be very similar phenotypically, that they maybe shouldn't be able to coexist as often as unrelated pairs. But at the same time, everyone here at the evolution meetings realizes that there's an evolutionary component to this question as well. And so we can consider how often should close relatives co-occur, depending on whether speciation was sympatric or allopatric, and how reproductively isolated they are, and most importantly, how much they can reproductively interfere with one another and not coexist for that reason. Well, for the most part, speciation tends to be allopatric. Of course, there are some key exceptions to this. But what should happen in the course of allopatric speciation, when the formation of completely independent biological species, is the development concurrently of strong reproductive isolation that eventually becomes complete. But at the same time, if this is happening in very distant sites, we'll get divergence in the niche and local adaptation to particular conditions. And so in order for close relatives that have evolved this way to coexist, there has to be some other event that brings them back together, an example of secondary contact. So some kind of dispersal that brings them back together, or an example of range expansion where they possibly can coexist in a small amount of space, or if the environment dramatically changes in a way that allows them to coexist. But there's reasons to think that this shouldn't really happen all that often. If we look at how reproductive isolation develops in this way, it's largely the case that reproductive isolation that forms in allopatry comes in the form of post-zygotic isolation, which is often very costly, because if you're coexisting with with another species and you have this late post-zygotic isolation, it can reduce fitness of both individuals, and that's what we call sort of reproductive interference. And we know very well from individual examples of what happens in secondary contact, we can have what's called reinforcement of the evolution of earlier barriers that can allow them to coexist and reduce this. But what we should expect is that if we see close relatives coexisting, they should have some of these pre-mating isolation barriers in addition to the post-zygotic ones. So in this talk, I'm going to show you a little bit of data where we asked a very straightforward question, and it's not what happens when close relatives co-occur, but across the landscape, how often should they co-occur? How often should we see them together? And so a simple way to sort of separate this out statistically is to look first at whether or not different pairs of species have distributions that overlap or do not overlap. Okay, so these are two spatial components. And within them, how are they associated, particularly associations within overlapping distributions? So the colors are each species, and these circles are sites. So think of that as a population that either has only one of the species, the other species, or a combination. And so we can have, if we start from right to left, completely disjunct, which we might expect if they've speciated in allopatry and they're ecogeographically isolated. Adjacent is an interesting one. It might be one where they have overlapped recently and there's been some competitive exclusion that puts them right next to each other but not together. Within the overlapping distributions, we can have them segregated or aggregated, and that's just compared to what we might expect simply by chance of how often that they would co-occur. And so what I would argue is based on both the arguments from ecology, this limited similarity hypothesis, and arguments based on reproductive isolation, is that we should more often find close relatives to be segregated rather than aggregated when they have overlapping distributions. And I won't go through all of this, but these are some of the reasons that you might expect close relatives, for example, to have more competition, more negative biotic interactions, despite the fact that they might have high niche overlap. This is the arguments from the ecological mechanisms. And then from evolution, we might again expect there to be some post-zygotic isolation, but in order for them to be aggregated or coexisting more often than by chance, we'd have to have these additional pre-zygotic. And so what we did is we did a couple of analyses. The first one was based on a published study. This is not our data. We took this data from a paper where they were looking at alpine meadows in the Swiss Alps. And this is at a fairly small scale. So they went out across about a 700 kilometer square area and looked in small plots at 175 of the most frequent species. And they did these calculations of how often were these pairs of species segregated or aggregated or just randomly associated. And we were interested in this because they happened to have congeners from 34 different genera in their data set. And so what we did is we redid their analysis, we got exactly their original result, and then we separated out to see what's the difference between unrelated pairs and congener pairs. And what we found is just a very simple overview of the results is if we look at so all of the species pairs had overlapping distributions because this was a pretty small total geographic area. But there was almost twice as many aggregated pairs when we looked at the congeners than when we looked at unrelated pairs. Okay, it's not a huge dramatic difference, but it's actually significant if you look at the non-random fraction. So this is interesting because, as I said at the beginning, we might expect them to be more often segregated, and here we're seeing that they're more often aggregated. And so to try to understand maybe a little bit more about what could be contributing to this aggregation, we're looking within a single genus. And so the study system is North American Lobelias. This is a single section in the Campanulaceae. You can see that it's got about 24 species, quite variable. They're all native to eastern North America. They do occur in a wide variety of wet habitats with a few exceptions, but for the most part, any wet, dirty ditch is going to be just fine. They're going to love it. They have a really high degree of species range overlap and sympatry. So we've actually done our own sampling, and there are individual counties that have 11 of these species. That's almost half of the clade in single counties. That happens twice, and they're not the same 11 species. And so there's a really high degree of overlap and coexistence here. We've done our own fieldwork and found 2 to 4 co-flowering species at about 10% of the sites that we found. So there's just a lot of overlap. And here's just some examples of a couple of species pairs. This is 2 species here, Pluvialula and Carnalis. And these are a little harder to see, but there's Georgiana and Nutalia, so it does happen quite often. All right, so a couple of interesting points related to this as we're looking at it. They also have a diversity of reproductive strategies that may be related to pre-mating isolation and coexistence in these sites. So we've had multiple shifts to annuality, polyploidy, obligate selfing, bird pollination, different sexual systems. So are these things associated with their pattern of coexistence within counties? And then I also want to point out that we didn't do this either. This was a study that was published in the 1960s. Some very diligent person made 191 species pairs crosses. It took them like 10 years. More than 85% of those hybridizations failed, so there's a lot of post-zygotic isolation here, so that's kind of important. So again, just to remind you, what we're going to do here is we're going to look at this lobelia data, look at their patterns of overlap at the county level, because that's a reliable data set, and then we're going to see whether or not these, where they fall in this distribution of spatial associations. Okay, so we did have county occurrence records, and that's a reasonable unit for the large continental scale that we're working on. It's also the most reliable data set of occurrence that we have. But we did quality control about 8,000 specimen records so that we could also look at overlap in flowering periods that we have inferred from these specimens. As I mentioned, we did our own field work. We have found a number of natural populations. We sampled from these populations in order to confirm ploidy among these different species. We grew them in the greenhouse to see capacity for autogamous selfing, and we're using mainly flower color to infer differences in pollination syndrome. I will say that there's some variation in flower size that's pretty dramatic, but we're not going to assume anything at this point based on flower size. So what we did for each of the species is to use these specimen records and infer a geographic range using alpha hulls. There's various ways to do this, but you can see that there's quite a bit of overlap among these species, particularly in the south, which is fairly typical for eastern North America. This is sort of maybe a legacy of glaciation and movement down there. So we have the ranges for each of these species, and what we did for this particular analysis was to take each pair and look at the region where their 2 ranges actually overlap, and then do county counting. We counted counties. And so we looked at where one of the species or the other species occur and where they were together. And this is a fairly conservative estimate of how aggregated they are, because you imagine this is presence data, and so you're likely to have one missing. So even when they do co-occur, it's likely to be a bit biased towards them being segregated rather than aggregated. And then just really quickly, we used the date of the specimen to get overlap in flowering curves to look at temporal isolation. Okay, I'm just going to jump right to the same kind of plot I showed you before. And again, we're looking at a much larger scale than we had for the previous data set. And so we did have several, about a third of them have non-overlapping distributions. Okay, so most of these are disjunct. These are candidates to dig in and see if they are eco-geographically isolated because they have very different niches. This looks like a small number, but this is 9 pairs where one species is in one county and it's not in the adjacent county, it's a different species. To me, that's a bit remarkable, but again, these are candidates for cases where perhaps there's been some competitive exclusion, and this is what's left. But what I like to focus on is digging into the data where we do have overlap in range between pairs of species, and showing again that even though we expect there might be good reasons for these pairs to be segregated in these regions, they're actually highly aggregated. There is a lot of overlap, okay? So most of the non-random pairs are aggregated. This is an index of spatial association, so we can actually quantify how aggregated and how segregated they are. And you can see that the majority are aggregated and some are very strongly aggregated. Okay, so just a little bit of a taste of some of the associations of this that we're looking at. So what do these aggregated ones look like? What's different about these pairs than the ones that are segregated? So we did a cluster analysis. This is not a phylogeny, this is spatial. So when you see pairs or clusters or groups here, these are ones that occur more often with each other than with any other species, okay? And so the farther they are down here, the more aggregated they are. So just quickly, the ones that are more segregated and tend to be pretty lonely are the ones that have very unique habitat types, right? So these are endemics to cedar glades, they're emergent aquatics, very small, so they have very unique habitats and they tend to be isolated. The ones that are aggregated, one thing that jumped out first to us is that these pairs all differ in ploidy, so they're all pairs of diploid, polyploid, which is interesting to figure out whether, there are others, pre-mating barriers to these, but it seems like that's a pretty good way that they could co-occur. In some of these, there's diploids only, but what we see is that in each case, there's either a shift in the flowering time, shown here in orange, a movement to complete selfing, or shift to bird pollination. So if you look closely at this particular group of 4, this is a cluster of 4, and in order for them to co-occur frequently, they each have to have some different kind of pre-mating barrier, right? Which is kind of interesting, and they overlap over quite a large range. But another pair, where they do also overlap over quite a large range, are very segregated because these are both diploid, bee-pollinated, outcrossing, nothing to really keep them apart except that they just do. They're just segregated. And given that this is a quantitative assessment of how aggregated or segregated these are, we can actually calculate using standard formulas for total reproductive isolation with pre-mating barriers, and there's a significant correlation between the degree of aggregation and the degree of pre-mating isolation when we do it as a matrix correlation. So just to sum up, what I've shown you here is that even though we might expect, both based on ecological and evolutionary hypotheses, that they should be more segregated, congeners appear to be, in many cases, more aggregated, and that this aggregation in the lobelias is associated with patterns of pre-mating isolation. So there's quite a few things we'd like to follow up on. I don't have time to go into them. We're really excited about figuring out what's going on here, and I appreciate your attention. Thank you.